Aloha everyone, and welcome back to Anatomy 151 here at Chaminade University. Today we're going to discuss Unit 10, which talks about muscle tissues. So the purpose of this entire lecture is going to be talking about the structure and the function of the three major types of muscular tissue, which would be skeletal muscle, cardiac muscle, and smooth muscle. And then we'll discuss the events that happen biochemically at the neuromuscular junction and describe how muscle cells are able to consume and utilize energy from their environment in order to make the cellular contractions. And then last but not least, we'll talk about the different types of muscle tension and how it is controlled. So again, there's three major types of muscular tissue. The first type is skeletal muscle. Next, we have cardiac muscle, and last but not least, we have smooth muscle. So the three major types of muscular tissue differ in a couple different ways. They differ in location, they differ in function, appearance, and also in control. So the skeletal muscle obviously is found at the skeleton, right? Cardiac muscle is found in the heart and the heart only. Visceral muscle is any of your internal organs. So picture, for example, your gastrointestinal tract. The purpose of skeletal muscle is to move bones. So the muscles that are attached to the skeleton move the skeleton, which allows your body to have locomotion. The muscle from the cardiac from the cardiac muscle has the purpose of pumping blood throughout the body and serving the cardiovascular system. And the visceral smooth muscle that's found in your abdominal organs, for example, perform various functions. But an example of this, given that we just used the gastrointestinal tract, is the process of peristalsis, or the churning of the food in your intestines and propelling it forward. So skeletal muscle has multinuclei. What that means is that we have more than one nuclei per cell that you're able to see, and that's because these muscle cells are going to need a ton of energy. These are going to be some of the most energy consumption cells in the body, and they also are striated, and we'll talk about what that means when we look at the actual histology. Cardiac cells are also striated, but they're, instead of being multinucleated, they have a single nucleus per, and they also have an additional morphology that's called intercalated discs. Again, we'll look at that when we check out the histology. Visceral muscle is different in that it has a single nucleus, so it's a mononucleated, and it has no striations at all. So it's easy to tell from cardiac and skeletal muscle, because if you have striations, you are dealing with cardiac or skeletal, but if you don't have striations, you're knowing that you're dealing with visceral or smooth muscle. Skeletal muscle is underneath our control. It's voluntary. That means that we can choose to pick up the pencil, for example, or type in the laptop. Cardiac muscle, on the other hand, is involuntary, although it does have regulatory control. So your heart rate can increase or decrease, but it's not something that you personally have voluntary control over. Same thing with your visceral muscle. It's involuntary. Again, it can be increased or decreased depending on different body functions, but it's not something that you as the individual are able to have conscious control over. So first we're going to talk about skeletal muscle. We'll talk about skeletal muscle tissue and the components of it. And here we have taken a transverse plane. And if we take a look at that, this is the bone clearly from this transverse plane. That bone would be the humerus, right? Here we have a tendon that connects the muscle to the bone. This is the first time I'm going to make this differentiation, but tendons connect muscles to bones, whereas ligaments connect bones to bones. So you're going to see that quite a lot as we go through some of the other sets of muscle attachments. All of the muscles are going to be attached to the bones by a tendon. All right, and as you might remember, the periosteum is the area that surrounds the bone, so that's that top layer of the bone. And if we're going to take a look at the muscle, first we're going to look at the whole muscle, but really we're going to take a cross-section looking at this fasciole, then we'll look specifically at that fasciole, then we'll take a look at the muscle fiber and look at that from there. So if we're looking at the biggest level, which would be the entire cross-section of the muscle, surrounding the muscle, we have this is called the belly of the skeletal muscle, and then surrounding the muscle, we have this periosteum region, which is going to surround every single muscle fiber or muscle cell, right? We also have a region here. This is the epimysium. It's going to surround this whole area, right? So it's going to surround the entire belly of the skeletal muscle here, and then as we go into this muscle fiber, that's also going to be surrounded by tissue. Again, if this outer layer here is the epimysium, the one that surrounds this muscle fiber particularly is going to be the paramecium. So every single one of these muscle fibers is going to be surrounded by a paramecium, right? Every single one of these fascioles, a fasciole has multiple sets of muscle fibers inside of it. So every single fasciole is surrounded by the paramecium. Inside that fasciole, we're going to have a ton of the muscle fibers themselves. Those muscle fiber cells are also going to have regions inside that have myofibrils if we continue to go down. All right, so here we've taken the fasciole, taken a cross section, doop doop, and we are now looking at what happens in a single, well, inside the fasciole we have, again, that's surrounded by the perimysium. And then inside here we have one particular muscle fiber. This muscle fiber has a ton of different things. One, it's multinucleated, so here's a nucleus here, here's another nucleus depicted there. You can also see it's innervated, that's going to be depicted here by the yellow lines, and then this is a blood capillary, so it's going to have a blood supply, and it has nervous supply to every single one of our myofibril, or our muscle fibers. Inside the muscle fiber is composed of many myofibrils, which are then composed themselves of filaments. Right? Every myofibril is going to have a sarcoplasm that surrounds it, and that's going to have the sarcolemma that surrounds the entire muscle fiber. All right, so I think I've touched on everything there. Let's going to continue forward. One of the other things that I'd like you to understand is just the basic concept. I won't go into the overview. We'll only go into the overview of how muscles are formed. I won't go into details. But essentially, you have muscle precursor cells. Those are these myoblasts depicted here. And the myoblasts start to associate with one another and line up and then start to fuse together. So that's how we end up with the multinucleation. Each cell originally had one nuclei, and then because they fused together to become a long muscle fiber, this was immature, but it's eventually going to become a muscle tissue, muscle fiber, and that's how we're going to have multiple sets of nuclei per muscle fiber. Additionally, we're going to have a remainder set of cells. These are satellite cells. I haven't talked about these yet, but satellite cells go on the outside of your muscle tissue, and these are going to be cells that are kind of like nurse cells that are going to help the muscle fibers and actually perform their functions by helping to feed them, etc., like nurse cells. All right, so if we're looking at the level of fasciole, right? Again, the fasciole is going to have the perimysium around the whole outside. Additionally, we have these satellite cells that I said are going to be found on the outside of the muscle fiber cells. Again, they're going to serve kind of as nurse cells to kind of help out. We'll talk about them a little bit later on. Inside a muscle fiber, which is what's depicted here in detail, but inside a muscle fiber, we're going to have several things, including mitochondria, etc. And each of the, and myofibrils, obviously those are the most important things. The myofibrils and the mitochondria are going to be providing energy. And remember, these cells are going to be constantly consuming a ton of energy, and so they're going to need a lot of assistance. That's why they have a ton of mitochondria. They also have a ton of nuclei, etc. Let's see. Surrounding each of these muscle fiber cells, we have the endomysium. So surrounding the whole, this entire fasciole, we have the perimysium. Surrounding each of the muscle fibers, we have the endomysium, and we also have the sarcoplasm and the sarcolemma. So the sarcoplasm is this region that's inside the muscle fiber itself, and if we look down here, we'll talk a little bit more about that. So the sarcoplasm is basically the area that is directly outside the muscle fibers themselves, and then we also have the sarcolemma, which is going to surround the entire muscle fiber. All right, so here let's take a look here. Here's a cross section of a muscle fiber, and again, the outside of that is going to be surrounded by the sarcolemma. That's here. Here we have multiple nuclei that are going to be surrounding this. Remember, it's a multinucleated cell, so here we have nuclei, nuclei, nuclei. Here's mitochondria that's going to be providing energy to the cell. If we take this sarcolemma and we take a look at it, it actually is going to run right along these myofibrils and connect almost directly into the sarcoplasmic reticulum, which is an area that's going to surround each one of the myofibrils. All right. We also, as we further look at the histology, are going to be looking at several different sections of the cells themselves. So you hear the word sarco several times. A sarcomere is something we're going to talk about kind of extensively. A sarcomere is going to be the region that is depicted from one region of the end of one part of the cell to another, where we have the contractility that occurs. And that occurs because we have these thin and thick filaments. The thick filaments are myosin and the thin filaments are actin. As you can see right now, this is a relaxed muscle tissue, so we don't have any contraction, and therefore we have space in between. But if we were to have it contracted, this actin head would be touching that actin head, and this myosin would be touching all the way at what's called the Z disc, or the edge of the sarcomere. We'll look at that a little bit more extensively here. So a sarcomere is one contractile unit of a myofibril, right? This sarcomere goes from one Z disc to the next Z disc, and It's going to include multiple different sections. The center of the sarcomere is called the M line, and it's called the M line because a ton of myosin molecules attach here. That's that thick filament here. So you can see that the myosin runs right along, and the M line runs right in the center of the myosin thick filaments. Those thick filaments have a partial overlap during a state of relaxation with the thin filaments of actin, which are connected to the Z disc. Okay, so the Z disc is on either side, the M line on the middle. Z disc connects directly to actin, M line connects directly to myosin. What does that look like? So it's kind of an overview. So here's that M line. Again, we're connecting directly to the myosin. These myosins are going to have these little heads on here. We'll talk about how they interact with the actin in just a minute. Again, the actin is connected directly to the Z disc right here. Now we also have these connecting filaments here. These are called titin filaments. Titin filaments indirectly connect the Z disc to the myosin, and that allows to keep the whole orderly arrangement, right? And so we have this zone of overlap here, which is basically a region where we have both myosin and actin when we are not contracted. When we have contraction, this is going to pull almost all the way to here, that'll pull almost all the way to there, and we will have almost entire overlap of actin and myosin. Okay, so the A band is considered the entire region that myosin takes up, so from one edge of the myosin thick filament to the other edge of the myosin thick filament. Again, that's connected by the M line. So the M line is directly in the middle of the A band. It's also in the middle of this H zone here. And the H zone is just considered the region where we have basically myosin by itself during relaxation. So there's no overlap in this region between myosin and actin. That's called the H zone. Then we have the zone of overlap. So H zone and the zone of overlap added together equal the A band, which is the entire length of the myosin molecule. And then that I band continues on, which goes all the way to the Z disc, and that's going to be the region where we have the I band is the region where it's just actin and also the titin filament, but we have no myosin overlap during a state of relaxation. That's going to change, of course, once we end up contracting. So again, this is just telling you what the components are and the description. So the Z discs, as we talked about, those are at the very edge of those sarcomere regions, right? And the Z disc is going to connect directly to the actin, which is here, and the M line is going to connect directly to the myosin. The H zone is the region where we only have myosin and we have no overlap. We also have the overlapping zone here. The overlapping zone and the H zone contribute to the A band, which is the entire length of the myosin molecule. And then the Z disc, which is found at the end of the sarcomere but at the middle of the I band, because the I band is where we have just actin by itself, no overlap with the myosin. Of course, there are titin filaments here as well. And so that's going to tell you which each of these are. So if you wanted to come back to this after you've done this entire lecture, just to refresh your memory and make sure that you know which piece is which, this would be a really good springboard for, say, a review section. All right, so let's talk about some of the different proteins that are involved in muscles and in their contraction and their regulation, etc. So the two major contractile proteins are myosin and actin. I've kind of already touched on those. Remember, myosin is a thick filament. It's connected at that M line. And then actin is going to be the thin filament connected to the Z disc, and there's a little bit of overlap. The regulation is going to be basically on the areas of actin where myosin is going to interact, and we've got two different regulatory molecules. One is called troponin and one is called tropomyosin, and they both are basically going to be involved in blocking the binding sites during the state of relaxation, and then it will open up so that it can bind during the state of contraction. We also have some structural proteins like titin. Remember, titin is going to run also to that from the edge here. Let me run back to the edge of the Z disc here over to where we're going to be interacting with myosin at the edge of that A band and also that direction. But it's not involved in contraction, it's more just structural. It's going to run the entire length of where actin runs basically by itself, because actin's a thin filament, and so it's going to help with structural support with actin during state of relaxation. We also have some other structural proteins, including nebulin, actin, myomycin, and dystrophin. You might recognize this from muscular dystrophy, so if there's some issues with this particular structural protein, you can end up with muscle atrophy. That's characteristic of that disorder. All right, so how does it work? How do we have muscles contract? And that basically happens by sliding. So basically myosin attaches to actin and then pulls. And when it pulls the thin filament inward. So myosin stays in place and the thin filament actin slides along underneath the myosin, causing contraction. And that means that the Z discs come towards each other and the sarcomere region shortens. The entire sarcomere gets shorter. And we have structural proteins inside there as well that are going to help transmit that force through the entire muscle. So the whole muscle is going to contract all at once, essentially. So this is the difference between relaxed, partially contracted, and maximally contracted muscle. So relaxed muscle, you have a large region, what's called that I band. That I band and the H zone are both going to shrink and then disappear when the muscle is entirely contracted. Remember that I band is regions where we have just actin, and that H zone is regions where we have just myosin. So it makes sense that as we shuffle inward from one direction, we lose both at the same time. So as this actin slides underneath the myosin, getting closer to that M line, and therefore this myosin is going to get closer to the Z disc, right, depicted here, and then eventually fully contracted here. We're going to lose regions where we only have myosin. At the same time, we're going to lose regions where we only have actin. So this is what fully contracted muscle looks like, and you can see that the entire sarcomere is drastically shortened, because now what we previously had is open space where we just had actin and just had myosin. Now we have full overlap between those regions. All right, how does contraction occur? Okay, so here we have myosin, and myosin has these heads that are these kind of hang off the edge, and they're going to have binding sites that are going to be attracted to the actin molecule, okay? But in order for that to happen, we have to have ATP. So let's start here. First, we have myosin heads that hydrolyze the ATP, and when that happens, so ATP then gets hydrolyzed, and now we have energy that's transferred. So when we break down ATP, that means that it releases energy. That release of energy then can be used to form binding and drive reactions. In this case, it drives the reaction of myosin binding to actin, forming a cross bridge. This is a very strong cross bridge, but it's temporary because it's going to actually contract and relax and contract and relax, right? But anyway, these cross bridges, once they are compiled, right, so once myosin heads are attracted to and cross bridged or linked to actin, then they can rotate towards the sarcomere. Can you see this head kind of is going to rotate downward and then over? And then they're going to detach and find another spot. And so they're going to pull basically inward until they get full contraction. So the contraction cycle is going to continue as long as we have ATP available and calcium. And I didn't talk about calcium yet, but you can see calcium here is depicted as purple, and it's going to be very vital. It's going to bind to different regions on the actin molecule. It's going to be vital for this contraction cycle. So as long as we have both ATP and calcium, this contraction cycle can continue. So what does that mean? That means in order for us to stop this contraction cycle, we're going to have control of those calcium channels, right? So all of it has to do with what's called an action potential. So we'll get there in a second, but let's go back to the sliding filament mechanism that I was talking about previously. So we have the thin filament, that's actin, right? And actin by itself is depicted here. It has two binding or regulatory proteins on it, troponin and tropomyosin, both of which, when they are attached, prevent the binding of myosin. Now, when calcium binds to troponin, that changes the shape of troponin and the tropomyosin complex, which then opens up a site on actin. And as soon as that site is opened up, then myosin jumps right on in, and now it can bind. Once myosin binds, then it induces contraction by binding and pulling until myosin is going to reach the Z disc here, and actin reaches very close to, if not entirely, into that M band. That's the full cycle of contraction. And in order for us to stop that, we're going to have to eliminate calcium. So at the top here, we have the sarcolemma. That's that region outside of that myofibril. We also have transverse tubules that run along that. These are cross-section, but if you picture, they're running into and out of your computer screen. And then we also have these regions on the sarcoplasmic reticulum, which was, again, the running down the entire myofibril. And they have what's called terminal cisterns that are able to store and release calcium. So when the channels are closed, the calcium is stored inside the terminal cistern. That means that troponin is connected to actin. That means that actin cannot bind to myosin, and we have relaxation. However, when we release that calcium, because of what's called an action potential, which is then going to basically send electrical charge that's going to say, hey, release the channel, release the calcium through the calcium channels. Once the calcium release channels open and the cell is flooded with calcium, that's again going to cause binding to troponin, which changes the entire conformational shape of actin, which allows it to bind to myosin, which allows contraction to occur. All right, so that's called excitation-contraction coupling. All right, so we know that the force of the muscle contraction is dependent on the length of the sarcomeres prior to contraction, and that makes sense, right? Because the longer, the more region you have to contract. So the further you have to contract, the more strength that you have in that contraction. We also know that there's an optimal length. So at some point, you can end up overstretched, and then it's not going to work as effectively. So we have understretched, optimal length, and overstretched. And so there's basically a optimal region in which you can have the highest percentage of contractile strength starting from a resting sarcomere length. And I'm not going to test you on that. I just want you to understand that conceptually. Let's talk about the neuromuscular junction. We have a picture in a little bit that's going to help follow me or back me up on what I'm about to say, but there's a couple of new things that we're going to introduce here. We just talked about the calcium channels, and I said that they happen because of an action potential, and I said it was electrically charged, but I didn't exactly explain it. So we have voltage-gated calcium channels, and that means that they are responsive to a change in electrical energy. And so they are going to get excited, and when they do, they get excited, or if the electrical change occurs, they get excited. If they do get excited, that causes that influx of calcium that I was just talking about released from the sarcoplasmic reticulum, and that causes what's called exocytosis, or release of a neurotransmitter. Now, a neurotransmitter is a biochemical molecule that is going to cause a change in cells downstream. Now, what does it do? So this neurotransmitter binds to these sodium channels on the end plate. Again, I'll talk about this in the picture in a second, but by binding to that sodium channel, that causes an influx of sodium into the muscle. The influx of sodium, which is again a positively charged molecule, causes what's called a depolarization event. Now, depolarization basically means that we have a change in the polarity, in this case the electrical charge, on the membrane, and that depolarization is actually what that action potential is. So that action potential is going to fire all the way along the entire cell, and that when that depolarization event occurs, that causes that release of calcium, because we have these voltage-gated calcium channels, and when we have a change in electrical charge, that causes calcium release from the SR, and then eventually that neurotransmitter gets broken down and the series of events stops. So without these series of events, muscle contraction literally would not be possible. So what does that look like? So I previously told you that muscle tissue is highly innervated, right? So this is a nerve. But what I didn't really explain is that every single nerve actually has multiple different innervations that are going to innervate the same muscle or the same series of muscles as well. In this case, it's going to be innervating the same myofibril. So this axon comes down and innervates the same myofibril in several regions. And if we take a look here at what's called the synaptic end bulb, or the end region of that nerve, and how it lines up at what's called the neuromuscular junction, which occurs again at that sarcolemma region. The sarcolemma is what is going to surround the myofibril. And if we take a cross section here and blow it up, here's what that looks like. So this is the axon terminal. Again, the whole thing is a synaptic end plate. And at this region, we have synaptic vesicles that are going to be releasing a neurotransmitter. This neurotransmitter is actually called acetylcholine. So from this point forward, you won't see NT, you'll see ACH, which means neurotransmitter. It's a specific neurotransmitter called acetylcholine. And acetylcholine is going to be released by these synaptic vesicles, and then end up going out and into the sarcolemma, right? Or into these regions of the sarcolemma that have these little clefts that will then end up causing a change or a depolarization of the membrane, which then causes the release of calcium from the voltage-gated calcium channels. So if we take a cross section here, again of this this synaptic end bulb, here we have ACH released from the synaptic vesicles that comes out here into this cleft in the junctional fold, where it's going to get bound to, where it's going to bind to ACH receptors, which changes the sodium content by throwing in some sodium, which then ends up causing this muscle action potential to be produced and then transmitted. So it all has to do with electrical charge, and that's how depolarization, repolarization of these membranes is occurring, which is basically what action potential boils down to. All right, so this is an SEM. That's going to be a scanning electron microscopic image. Here we have a blood capillary, and here we have an axon. Now, unfortunately, it's not color-coded for you, but normally nervous tissue would be yellow and blood capillaries would be shown in purple, right? And here we have the somatic motor neuron, which connects directly to these axon terminals. The axon terminals have these synaptic end bulb regions, which is going to be releasing the neurotransmitter acetylcholine, and we're going to have calcium involved in this, and then that's also going to cause a sodium release, which is going to cause a change in the depolarization, or cause a depolarization, which is a change in the electrical charge of the membrane, and that's going to be what we call an action potential. Okay, so this is everything that I've told you, kind of all wrapped up into one. And it looks like a lot, but don't worry, I'm going to walk you through it. So let's start up here with one. One is when we get that electrical impulse. That's the nerve impulse that actually is going to be coming down from from the axon. Once that happens, we have a release of acetylcholine. Acetylcholine is going to be released by that synaptic envelope and go into these synaptic clefts where they bind to ACH or acetylcholine receptors. When that binding happens, so acetylcholine is going to diffuse across that synaptic cleft, bind into its receptors, and then trigger what's called an action potential. Remember that action potential is actually a depolarization in the membrane, so it's a change in the charge of the membrane that occurs, and that happens because we're flooding it with positive charges. But I digress. So basically that muscle action potential, which starts in one location, is going to travel all the way along the outside of the cell, whereas inside of the cell, acetylcholine coming in is going to cause that action potential to occur. And we have another enzyme here called acetylcholine esterase. Now, acetylcholine esterase is going to be something that's going to attack and degrade acetylcholine. And this means that we can only have one action potential at a time. We can't have a second action potential occurring until another set of acetylcholine is released from the motor neuron. So once acetylcholine is released, it does its job, it goes through, and then it gets digested or degraded by acetylcholine esterase. All right, now remember we just triggered that muscle action potential, which is going to be traveling here along the outside of the cell. Muscle action potential traveling along, it's going to go into these transverse tubules that we saw. Those tubules are going to open calcium channels. So the calcium release channels open in the sarcoplasmic reticulum, also known as the SR. And that's going to mean that calcium ions flood into the sarcoplasm. Once those calcium ions flood in, remember what they do? What do? They bind to the regions of troponin that are going to change the conformational shape of troponin and tropomyosin on the actin molecule, exposing regions for myosin molecules, the myosin heads, to bind to those actin binding sites. Once that happens in the presence of ATP, recall, and elevated calcium, we end up with what's called the power stroke. That contractile event, which again uses ATP, where the myosin heads bind to actin, pull in, swivel, release over and over until they contract all the way in until the sarcomeres shorten. Right? Now, the end of this cycle is that the calcium has to get back into the sarcoplasmic reticulum, right? It has to go back in here. And how does that happen? So what happens is we have these active transport pumps that are going to be pumping calcium back in through the, and at the same time that calcium release channels are going to close. This is all using ATP as well, mind you, so this is going to be very expensive for the cell. That's why these cells need a ton of energy, lots of mitochondria. But what happens after this is that we end up with the reduction in calcium in the sarcoplasm because it's all gone into these regions and is now out of the sarcoplasm. That means that we now are going to have troponin binding and tropomyosin binding back to actin, so it slides back into place, preventing the binding of myosin, which then relaxes the muscle, right? And this is basically a lather, rinse, repeat if you end up with the secretion of acetylcholine, which is going to cause this action potential, right? So how do we get the energy necessary? Where do you get that ATP from to drive that power cycle, right? So first we have creatine phosphate, then we have anaerobic glycolysis, and we also have cellular respiration. So these are all ways in which we can create energy for the muscle cell. Now, creatine phosphate is the very first burst of energy that a muscle cell can get. Basically what happens is ATP is going to drop off its phosphate at creatine phosphate during relaxation, and then that ADP molecule can go find another phosphate and do this over and over and over, creating a storage in the cell of creatine phosphate that we can use as an immediate source of energy. When muscles contract the very first time, we can then offload that phosphate very quickly from ADP in the sarcoplasm, which then can become ATP and high energy over and over and over. So basically this is a way that we can have a storage of an extra phosphate very quickly to make ADP ATP for about 15 seconds worth of energy for a muscle contraction. Again, that is ATP that is actually going to be getting the phosphate from creatine phosphate. Now if we want a little bit longer energy, we can get something from anaerobic glycolysis. And I'm not sure if we've gone too much into the cycle of glucose breakdown, but if we do it without oxygen, meaning that we build up lactic acid in our blood. This is why we end up, sometimes if we get cramping, that's because of a high amount of lactic acid that ends up, or Charlie horse, that ends up building up in the muscle. And that's because when we're doing anaerobic glycolysis, we are creating ATP from the breakdown of glycogen into glucose. Basically, the idea behind cellular respiration is that you take glucose, you break it down and get energy out of it, and the resulting byproducts are going to be water and oxygen. But here, if you do it without oxygen, you're going to be getting only 2 ATP per glucose molecule. If you do it with oxygen, you get 34 or 36 ATP. So it's almost tenfold more to be doing to be able to take glucose and break it down to be able to undergo the process of cellular respiration and get ATP out of it. However, that takes time. So that takes at least a couple minutes to get started, but it gives you a lot longer source of energy. So you're able to have that for several minutes to hours. And the cool thing is that you can actually get you can get glucose from a lot of different places. You can get it from amino acids that get transferred over. You can get it from fatty acids in your fat cells, from glycolysis, breaking down pyruvic acid, which here is this next part of the glucose breakdown chain, and all of that happens in the mitochondria. The mitochondria are what creates that energy, lots of ATP out of the breakdown of glucose. So the very first burst of energy comes from creatine phosphate, which is going to feed the creation of ATP. The second burst of energy comes from anaerobic glycolysis, but it's inefficient, although it's faster. And then later on, after several minutes, you're able to have a much higher energy source from every single sugar molecule. But again, it's going to take a little bit longer. And so this is going to be several minutes to hours. It's something like when a marathon runner is going to carb load so that the next day they have a longer source of energy. And they basically are taking those carbohydrates, like pastas or whatever, and they're breaking them down into glucose, and then the mitochondria are breaking them down even further by the process of glycolysis into a lot of ATP that they can use for long duration of energy. All right, so what happens when you just simply run out of strength, right? What is muscle fatigue? And you can eventually get your muscles to the point where they're simply unable to maintain that force of contraction after prolonged activity. And there's a couple different steps that can kind of go wrong in these pathways that can lead to muscle fatigue. The first is that you can have an inefficient amount or an inadequate release of calcium from the sarcoplasmic reticulum. So you can have low amounts of calcium to begin with, or you can just have not enough calcium released at the right time frame. You can have a depletion of creatine phosphate, for example, oxygen, other nutrients that are needed to be able to make that ATP, right, that have to go into the cycle, the Krebs cycle. Additionally, if you're using anaerobic respiration, you can get a buildup of lactic acid, or you can have an insufficient release of your neurotransmitter, acetylcholine, at the neuromuscular junctions. All right. Ever notice that you exercise and then all of a sudden you start to pant? You breathe very heavily for a little while, even after you've stopped that exercise, and that's because you are in oxygen debt at this point, which means that you have to breathe heavily for a little while to replenish that oxygen stores. You also have to, again, reload that oxygen onto your myoglobin and replenish your creatine phosphate, and you have to take lactate and convert it into pyruvate, otherwise you have lactic acid buildup. So we have to make sure that we have a lot of oxygen on hand after exercise to be able to complete these activities. All right, so let's talk about control of muscle tension, right? You don't just contract a muscle or relax a muscle, particularly if you're doing something like standing or walking or even just holding a pencil, right? You're moving it in a precise fashion. You're contracting it not a lot, not a little, but somewhere in between. And the strength of that contraction depends on several things. First, it depends on how many motor units are activated. It depends on if you have already just recently contracted. So you have several different stages to contraction period. So let's talk about twitch contraction. How do your muscle fibers all respond together to a single action potential? All right, so we have several different points here. Let me just walk you through this graph. This is the force of contraction, so obviously stronger, weaker. And this is the time in milliseconds from point 0. So we have a latent period, which is when we have received a signal, like acetylcholine, but we haven't yet had a release, etc., that's going to lead to, like a release of calcium that's going to lead to contraction. Then we have a contractile period. And then after we have a contractile period, we have a relaxation period. Then we have a little bit of a lag before we have the latent period as well, and that's called the refractory period, a period of time in which we're kind of unable to get the next action potential going until the stage is reset, essentially. All right, so eventually the waves are going to add up or summate. So wave summation occurs when a second action potential triggers muscle contraction before the first contraction is finished. So if you start to contract and then you get another signal to contract again, and then you get another signal to contract again. Eventually, you end up with full contraction. So this is going to result in a stronger contraction over time. And we have two different types of the stronger contraction. The stronger contraction is called tetanus. We can have an unfused tetanus or a fused tetanus. What does that mean? Okay, so here we have a single twitch. This means that you've gotten one action potential. So one action potential comes in, that action potential leads to release of calcium, that calcium then binds to troponin, which changes the actin, which makes the actin bind to myosin, which allows for a little bit of a contractile event. Eventually that contractile event is going to end up relaxing and getting a latent period. Now if we have two action potentials that occur back to back, that's when wave summation occurs. We've added them together. So here we have the first action potential, then we get a secondary action potential just before the contraction period has ended and entered into the relaxation state, which is shown in black. So this is shown basically what would normally happen here. So now we have a secondary action potential that continues that contraction event, and then a relaxation period. Now if we have several of them that come together that are going to give that constant getting just a little bit weaker and then stronger, and then a little bit weaker and then stronger, kind of like this, that's called unfused tetanus. It's a series of action potentials that are timed in such a fashion that we're just starting to relax right before we're told to contract again. So we end up with a stronger series of contractions than we would have had by just one action potential, and that's called unfused tetanus. And fused tetanus is when it's just constantly going, so we never even get the chance to come back down that slope at all. We're just going to enter straight up into the top amount of the force of contraction until we hit muscle fatigue, and then we're going to end up here. And that's why they tell you to get your tetanus shot, or the disease that's called lockjaw that can occur if you step on a rusty nail, for example, because you get an infection. And that's because this is what it induces. The lockjaw is basically because you're forced to contract. You're in a fused tetanus state for whatever muscles are constantly firing. And so that's what goes apparent in someone who acquires a disease called tetanus. All right, so how do we recruit motor units? So the weakest motor units are the first ones that are going to be recruited, and then the stronger motor units get recruited later. And that's very important for very fine motor control. So if you were, for example, to try to pick up a grain of rice, you would want your very weakest motor units to be working at first, so that you're very careful about what you're doing until you get to the point where you need more strength, and then you'd have the stronger motor units do something that would be more brute force. Now, we also have motor units alternately contracting. Therefore, contractions are able to be sustained for longer periods of time. We don't have any strain on any particular cell or any particular motor unit over the others. Now, even when we're at rest, your skeletal muscle exhibits a small amount of tension. That's called your muscle tone. And this is a measure of physical fitness. And this is basically due to involuntary contraction of the motor units while you are at rest. Now let's go back to how we lift or lower something. So for example, if you have a pencil and you want to put it on the table, you don't just drop it on the table, you lower it down to the table. Or if you want to pick it up, you don't toss it in the air, right? You pick it up very slowly with careful control. And that's going to be under the force of isotonic strength. And isotonic strength is when the tension is constant, but the muscle length changes. And the change in muscle length is, of course, what moves the bone. And we have two different methods of this. One's concentric and one's eccentric. Basically, if you are going to be picking up a book slowly, and the movement means that the muscle is slowly contracting, it's slowly getting shorter over time, you have the same tension, but you're decreasing, the muscle length, so you're increasing the contraction, right? That's called concentric. And if you're relaxing a little bit, you're keeping the same muscle strength, I apologize, but you are expanding the muscle a little bit, it's getting longer, that's going to be eccentric contraction. Now, if you were to hold that book out at arm's length and not move at all, that's called isometric contraction. And isometric contraction is when the muscle contracts, but it doesn't change length. So you hold it at the same contractile tension and at the same length for whatever it is that you're doing, whatever activity. All right, we also have multiple different types of muscle fiber types. And if we were in the lab, I'd be showing you this in histology, but you can see here under a light microscope that we have major differences in morphology between what's called slow oxidative and fast oxidative glycolytic versus fast glycolytic fibers. And basically what this means is, do they respond slowly or quickly? And how quickly do they burn through their energy, right? So obviously the fast glycolytic fibers are going to be the first ones to respond. And then these are oxidative and glycolytic. And then the slow ones are oxidative. And so we have the reason that they respond more quickly or more slowly is their mechanism. If they're oxidative, they're slower to respond. If they're glycolytic, they're going to be faster to respond. And then these are kind of like the intermediates. All right, so there's multiple different types of skeletal muscle fibers. Additionally, you can increase your muscle tone by stretching and core training, etc., and strength training. All right, so let's talk about cardiac muscle. We're going to kind of shift gears. We've been talking heavily about skeletal muscle. Cardiac muscle is very similar to skeletal muscle in terms of the arrangement, but it has a major difference. It has what's called intercalated discs, and those are depicted here, right? They look like that. So it still has the striations that you're used to for your skeletal muscle, but it also has intercalated discs. So it's a very easy thing to be able to depict histologically. Smooth muscle is a little bit different. First of all, smooth muscle is not striated. Secondarily, smooth muscle contractions are going to be a little bit slower and last longer. They're going to be kind of a little more constant as opposed to something like skeletal muscle. You pick up the pencil, you put the pencil down, you're finished. Whereas something like your uterus or your bladder or your gastrointestinal tract constantly working. So the contractions start more slowly, but they last longer than both skeletal and cardiac muscle contractions. And smooth muscle can shorten and stretch to a much greater extent than cardiac or skeletal muscle. So smooth muscle is made up of a series of, so smooth muscle tissue is visceral or single unit, and it's made up of a series of individual cells. There's a multi-unit smooth muscle tissue. Again, anytime that you see orange, you're going to see innervation, and you'll note that one nerve is going to innervate multiple cells, and that each cell is going to be innervated multiple times. This is going to allow for our muscles to coordinate with each other, so that all the muscle cells in a particular unit will be firing at the same time. However, again, it's multi-units, but these are going to be innervated by different nerves, so they might not all be activated at the same time, allowing for different contractile forces and different weak or strong motions, right? Like picking up a pencil has more dexterity than something like swinging a hammer. So if we're going to look at relaxed versus contracted smooth muscle fibers, so here's relaxed and here's contracted. Again, these are going to contract because of the actions of actin, which is the thin filament, and myosin, which is that thick filament here. And so you can see here it's relaxed because we don't have overlap, and you can see here that it's contracted. All the way we have overlap, we have a much shorter sarcomere. And then this region here is our dense body, which is basically going to be connecting multiple different sets of cells together. And that is what an overview of a smooth muscle fiber looks like. All right, so as muscle tissue gets older, particularly skeletal muscle tissue, they lose the ability to undergo mitosis. So as the skeletal muscle becomes completely mature, it can no longer undergo mitosis. They're actually going to be replenished by a series of stem cells, just like most cells in the body. And if they end up to the point where they're too old, they can have a couple different things that happen. We can have hypertrophy, we can have hyperplasia, and we can also have smooth muscle and parasites. So there's several things that can happen in terms of regeneration, but they're not able to. So they can get larger, right? Larger, and we can get more of them, but we're not going to be able to have mitosis occurring where one cell is making another cell. As you get older, approximately 30 to 50 years of age, you lose muscle tissue. Approximately 10% of the muscle tissue is slowly replaced by connective tissue and adipose tissue. And by the time you're 50 to 80, another 40% of our muscle tissue is replaced. Now, this is something you can stave off mildly with routine exercise, etc., but it is something that is going to be subjected to everyone as they get older. And that's going to have several consequences. First, you're going to have a slowing of your reflexes, and you're going to have an increase in the slower oxidative fibers as compared to the faster glycolytic fibers. You're also going to have a decrease in flexibility and muscle strength. All right, guys, so thank you so very much for making it through to the end of Chapter 10. I appreciate you joining us for class today. Aloha, happy studying, and I will see you next time for Unit 11.